In this lab, we're going to talk a little bit about blood. I didn't expect to really talk about this. We talked about a little bit as connective tissue, but I thought blood probably deserved a little bit more attention. And so I thought, let me just spend another couple minutes and tell you a little bit more about blood, which I think is sort of one of the interesting things. So hopefully you'll enjoy this, and it's going to be giving you a little bit more of an insight about the different types of blood cells, where they come from, and things like that. So let's talk about blood, okay? First of all, when we look at blood, okay, we know blood is made in the bone marrow. The bone marrow is what makes all our blood cells, whether it be a red cell, whether it be a white cell, or whether it be something we call platelets, which are things that are important in clotting. Okay, they all come from the bone marrow. Babies, almost all their bone marrow is able to produce blood cells, but as we get older, less and less of our bone marrow is able to do that. Most of our bone marrow gets replaced more with fat as compared to blood-forming cells. Okay, but the thing that's important to remember is that what happens is when we think about making blood from the bone marrow, they all come from this magic thing called the hematopoietic stem cell. Okay, and only about 1 to 2% of all the cells that are in the bone marrow are these hematopoietic stem cells. What a stem cell is a stem cell that doesn't know what it wants to be when it grows up. Okay, it's sort of like whatever way it wants to go. Okay, and whatever the body tells it to do, it says, yes, boss, I want to do that. Okay, and that's what this hematopoietic stem cell is. So this one individual cell, this hematopoietic stem cell, could become a platelet, could become a red blood cell, could be all these different types of white blood cells, and could become another white blood cell called a lymphocyte. Okay, and we'll talk about that in just a second. So let me get rid of those little those little scribblings I have here. What happens is under the right stimulus, depending upon what we need. Okay, under the right stimulus, what happens is hematopoietic stem cell says you can go one of two ways. Either go to the road to the left or the road to the left or to the right. The road to this way over here is called the myeloid stem cell. Okay, so that same hematopoietic stem cell could either become a myeloid stem cell or a lymphoid stem cell. Okay, and that's going to be sort of important because once they get to this stage, guess what? Their destiny is pretty much almost predetermined in some way. Okay, that myeloid stem cell then further differentiates. Now, we talked about differentiation before, and that's going from a very primitive cell going through multiple stages to become an adult grown-up cell. And what happens is myeloid stem cell produces what are called platelets. Now, platelets, like I mentioned before, they come from what are called megakaryocytes. These megakaryocytes are large cells that will break up into small little parts. And what these parts or platelets do is if we have a little hole in a blood vessel, what happens is those platelets go up and they create a little plug. They're sticky and they stick together. When people take aspirin, what aspirin does, and the reason why they tell people who are prone to having a stroke or a heart attack to take an aspirin today, is what the aspirin does is it decreases the stickiness of the platelets. So the platelets, instead of causing a platelet plug inside of a vessel, which then leads to more clotting, which causes more red blood cells to collect behind that platelet plug because of certain chemicals that those platelets will give off that will attract red blood cells to form a clot. To prevent that clot from forming, they take aspirin so the platelets don't start that first process of the platelets sticking together. Therefore, they can't release those chemicals. They can't attract red blood cells, and therefore the red blood cells don't make a clot. Okay, it's really that simple. Okay, so these platelets are helpful in clotting. They're the first basic step in clotting. The second type of stem cell that we get is called an erythrocyte. Okay, and the erythrocyte, erythro, if you're taking medical terminology, means red. That's my red cell. In the blood, per cubic millimeter, there are millions of red blood cells. Okay, I mean there's tons. Okay, it's the most populous type of cell in the blood. Okay, and what I should say is besides the erythrocyte, there's this thing right here which is called a reticulocyte. Okay, it's a reticulocyte. And we'll talk about red blood cells in a minute. We'll talk about reticulocytes. Reticulocytes are really important for another reason. I'll mention that in just a minute. Okay. We could also go from a myeloblast or a monoblast, and a myeloblast goes into these types of cells. They're called basophils, neutrophils, eosinophils. Now, these you know because what happens is when you look at them, we're going to talk about them in a little bit, they have granules in those. And those granules are little chemicals, they're in little containers, and these granules stain differently. Basophils stain with these little granules, they stain dark purple. Eosinophils eosinophils, okay, I'll go to eosinophils. Eosinophil means red. And what happens is eosinophils have these red granules, okay, and they have different chemicals that stain red. Neutrophils, guess what? They have granules, but they're very difficult to see because they're very neutral. You can't see them a whole lot, okay? We also have what are called monocytes. And monocytes are big eaters, okay? And we'll talk about what each of these cells do, what each of these white cells do. Monocytes, it's funny because when they talk about white blood cells, they say they're called granulocytes or agranulocytes. A means without granules and G means granules. They usually link or lump monocytes into agranulocytes, but monocytes actually have granules as well. You know why? Because they came from that same myeloid stem cell, okay? And we'll talk about what each of these white cells, red cells, and the platelets do a little bit more. What happens is this lymphoid cell line results in what's called a lymphoblast, and these lymphoblasts create what are called lymphocytes or NK lymphocytes or natural killer lymphocytes, okay? They don't have any crazy tattoos or anything like that, but they're called natural killer lymphocytes or other lymphocytes. And we have two types of lymphocytes. One's called a T lymphocyte and one's called a B lymphocyte. And I'll talk about those in a little bit. B lymphocytes, actually what they do is they actually go on and they become factories, they become plasma cells. They further go through another differentiation, they become what are called plasma cells, and plasma cells make antibodies from these plasma cells. From the antibodies are made. T lymphocytes are type of lymphocytes, but they sort of are eaters as well. Okay, and we'll talk about those in a little bit. So let's talk a little bit more about blood cells. When we look at blood, okay, blood. If you drew a tube of blood, okay, you've probably all given blood at some time, and the blood when it comes out will look something like that. It's sort of like a cranberry color. It's sort of darker red because it's venous blood. Venous blood doesn't have a whole lot of oxygen, and I'll talk about why that's like that, why it's that color as compared to bright red. But if I let that sit, what happens is the red blood cells, because there are a lot, and white blood cells because there are some, where the red blood cells are in the millions, white blood cells are in the thousands, okay? What happens is because they have weight, they fall, they sink to the bottom, okay? And they sediment. They start to float to the bottom, or not float, they start to sink to the bottom, and the plasma separates from that. So what happens is we have plasma that's on the top, which is like a straw-colored fluid, and the bottom base in here is basically all this down here would be cells, okay? Now that's what blood is comprised of. If we let it sit there, your blood has a liquid component, which is the plasma, and it's about 55% of your total blood volume, okay? What happens is the 45% are basically red cells, because again, they're so plentiful. There's in the millions per cubic millimeter, okay? So the red cells are about 45%. And then what happens is on the top of that, we have what's called a buffy coat. And that buffy coat is less than 1%. And those are the white blood cells and platelets. Why? Because they're in the thousands. The normal, we're talking about, 2 to 3 million red blood cells versus, 5 to 10,000 white blood cells per same volume. So in other words, actually more than the 2, 3. It's probably about, 4, up to maybe 4 million red blood cells versus, 5 to 10,000 white cells. So this is called a buffy coat right here. Now, what happens if I look at this percentage, which is basically the erythrocytes versus the plasma, okay? That's called the hematocrit. Hematocrit. This is a very important number. You're going to hear them if you're working in the hospital say, what's the hematocrit? Hematocrit tells me what the percentage of red blood cells is, because again, that buffy coat's negligible. It's a 1%, less than 1%. The percentage of the red blood cells versus my percentage of my plasma. Okay, and it's usually around anywhere between about, if you look here in the normal blood, it depends on males or females. Females are a little bit lower, males are a little higher. The hematocrit in females is about 37 to 47%, and in males is about 42 to about 52%. Okay, and that's what it's supposed to be. And if people have anemia, what happens is their hematocrit percentage will go down. Now all of a sudden we're about 35, now we're 32, now we're 30, now we're 25. And this is what we see with anemia. That hematocrit level goes down. Okay, so that's just looking at what we have here. Again, that plasma versus basically my red blood cells, because again, that buffy coat, very small amounts of white blood cells, platelets like that in that intermediate layer, which we actually see right there. Okay, and that's called the hematocrit. I think that's an important factor to remember. I think it's something that's interesting. I know when we looked at when I remember when I was doing part of my residency and doing an emergency room, a lot of times if you had trauma, what you needed to do is you're always drawing blood, but you might want to know the hematocrit. We used to actually take a little capillary tube and then get a little and suck up a little of the blood in the capillary tube, and then in the emergency room we had a centrifuge, so we stick these little capillary tubes in the centrifuge and spin it down, and it would actually cause the red blood cells, because again they have what? They have weight, to fall to the bottom of this capillary tube, put a little piece of clay in the bottom, okay? And then what we take is to take the capillary tube, put on this wheel, spin this wheel, and we're able to actually see where this line intersected, which was at a percentage, which tells us percentage is. So if in other words, if they had trauma and they were bleeding, what we'd find is that percentage instead of being, a 40% or 42, 45% in the male is now down to 35, and we'd follow it again, maybe down to 30 means they're still actively bleeding. Okay, so we look at that hematocrit to look at the amount of red blood cells that they really have, the percentage, because it probably should be somewhere around 45%. 45%. If you think 45%, that's probably a pretty good number, and that's called hematocrit. So that's the difference between the plasma on the top, which is a liquid, and compared to the red blood cells or erythrocytes, which are on the bottom. Now, this is just looking at something called a CBC. Okay, now I just should bring this out because if you're working in a hospital, you're going to hear this all the time. CBC. CBC stands for complete blood count. Okay, and what they do is they look at a bunch of factors. They look at the white blood cell count. They look at the number of white blood cells. They look at the red blood cells. They look at the hemoglobin and hematocrit. Hemoglobin, and we'll talk about this in a second. Hemoglobin is a large protein that's in red blood cells, and the hemoglobin basically is a boxy type of a molecule, and in the corners of it, it actually has iron, and the iron binds oxygen, so the oxygen could be carried around through the body. We'll talk more about this when we talked about the cardiovascular system a little bit later in the semester, okay? But we look at this is what this is. Also looks at something, this is called indices, MCV, MCH, and MCHC, and that just shows you the size of the red blood cell, the amount of, the concentration of hemoglobin in the red blood cell and stuff like that, okay? RDW is something that you don't have to remember these. I just think they're interesting to know in case you hear these down the road. RDW is they look at the small red blood cells and they look at the large red blood cells, and they sort of look at the difference between them. What percentage is different? Okay, and we find that in most people, the percentage, there's very little difference in the change of the red blood cell size. When I show you some red blood cells, you'll see that some look a little bit larger, some look a little bit smaller. But the RDW looks at basically, the difference in sizes. What's the percentage in difference in size? And that's called RDW. Certain types of anemias, they actually start to have very small red blood cells, and other types of anemias have a very large red blood cells. And in those cases, that RDW percentage will go up because there's a bigger difference in that size. Okay, we look at the platelet count. Platelet count, again, the platelets are important in regards to clotting. Okay, we're talking, when we talk at white blood cells, again, we're talking about thousands, thousands, where red blood cells, we're talking about millions, 4 to 6 million per about, 5 to 10,000 of white blood cells. Okay, hemoglobin is usually, somewhere around 15, and the hematocrit is probably about 45 usually, and somewhere in that range. And these don't you have to worry about those indices. And then we look at the platelets, we're talking in the hundreds of thousands. Okay, where we're talking in the 5 to 10,000 white blood cells, we're talking anywhere between 100 to 400,000 platelets, but they're very small. They're little particles. I'll show what they look like. Then this right here, down here, this is called the differential. Okay, when look at this, is called the differential. They usually sometimes call it the diff. Hey, what's the diff? And the diff just shows what the percentage is of each of the different white blood cells. Okay, what happens if you look down here? We talked that there's different types of white blood cells. Okay, and we talked about what are called segmented neutrophils. That's the most common type. We'll talk about those in just a minute. That's the most common type. Lymphocytes are the next most common. When we see a bacterial infection, this number of my segmented neutrophils goes way up. Okay, if I see a viral infection, I usually see that increase in my lymphocytes. Now, monocytes are small, 0 to 15%. Eosinophils are 0 to 6%, and basophils are even less than that, 0 to 2%. And they occur in different things. I'll talk about each of those individually. But this is called a differential. This whole test right here is called a CBC, but this down here is called the differential. And they just sort of divide. If they take 100 white blood cells randomly, what percentage of them are neutrophils, what percentage are lymphocytes, what percentage are monocytes, what percentage are eosinophils, what percentage are basophils. Okay, and that's where they go with that. Okay, and so that's just called a CBC. I think it's important to remember that name, and because it's something that you'll hear all the time in the hospital. It's something in a hospital and clinics. You'll hear CBC. You look at the white count, you look at the red count, you look at hemoglobin, hematocrit, and stuff like that. We know hematocrit's that percentage of red blood cells as compared to the plasma. Okay, and the hemoglobin is the amount, actually the amount in grams. The amount in grams of hemoglobin as a protein in red blood cells, and that hemoglobin is important. Again, in anemia, what we find is we have fewer red blood cells, so there's less amount of the hemoglobin per unit volume, so the hemoglobin levels will also go down in various types of anemias. Now, these are erythrocytes. If we look at the erythrocytes, again, this is looking underneath the microscope, and almost every cell you see, with the exception of this puppy right there, are all red blood cells. Okay, they're all red blood cells or erythrocytes. Because again, we're talking, 4 million red blood cells as compared to 5,000 white blood cells. Okay, so anyway, and you can actually, these things up there, it's a platelet right there. There's a platelet right there. There's a platelet right there. A couple of the little platelets are in there because again, those are in the hundreds of thousands. So those are called erythrocytes. Okay, and this is what a erythrocyte looks like. Now, I want you to just look at this, and this is, they look like those little candy that used to have those really crummy things that they're hard and they all taste the same and they're really sickeningly sweet. Okay, but what they're called is they're called biconcave discs. Biconcave discs. If I looked at this red blood cell from the side, it would look like this. And it's thin in the middle. That's where this is. That's where we see that the thinness, the thinness occurring in the middle, and where it looks like a little donut, but the hole's not totally out. That's because of this area right there. They're called biconcave discs. Now, one other thing that's important to think about red blood cells, okay, is that what happens is as they mature, as they go from that myeloid cell line, like we talked about earlier, and they continue to progress to become a mature red blood cell that looks like this, they go through something called a reticulocyte. Okay, now if you look at these red blood cells, what don't you see? You don't see a nucleus. As these develop, what they do is when they start to get closer to a reticulocyte, what they actually do is they break up the nucleus and they eventually get rid of it. They excrete it from the cell. And the reason why they do that is what happens is red blood cells have one purpose and one purpose only, and that purpose is to carry oxygen. So what they do is they say, the nucleus takes up too much space in here. Let's get rid of it. So they get rid of the nucleus so they could carry more oxygen in the red blood cell. And that's a good thing. That means that they can't divide or anything like that. They actually last about 100 to 120 days. And after 120 days, what happens is the spleen takes them, which is up and just next to your stomach in the left upper quadrant, which everybody should know about right now. The spleen takes them and sort of destroys them, but we reuse most of the stuff from the red blood cells. The iron we take from the red blood cells, we reuse it. We stick it back in the system and use it again. So, which is really sort of cool. And then what happens, this is also great, what happens is the protein that's left that starts to be degenerated and denatured what happens is the red blood cells, as they're being destroyed, what happens is a lot of the products get sent to the liver, and the liver makes something called, from the red blood cells that were the initial red blood cells, for the byproducts of them, to make something called bilirubin. And bilirubin is that stuff that sometimes when we have certain liver problems, the bilirubin levels increase in the blood, and what happens? That bilirubin is a yellowish brown pigment, and that's what gets in the skin, and it causes the skin to become sort of yellowish appearance with what we call jaundice. So the red blood cells don't have a nucleus. They last about 100, 120 days, and their sole purpose, or the largest purpose, there are a couple of minor purposes, but their sole purpose, because they could carry carbon dioxide and a couple other things as well as the oxygen. But their primary purpose, their major purpose, their goal in life is to be able to carry oxygen. And they've gotten rid of the nucleus simply because the nucleus just takes up too much space. They could carry more oxygen if they have more hemoglobin instead of a nucleus, and they do that. And those are what red blood cells look like. If we look at them underneath the microscope, that's what they look like. See how they look pale in the middle? They look darker around the outside and look pale in the middle. And the reason why they look pale in the middle is because they are biconcave discs. They're that disc that's like this, thin in the middle, and therefore, because that area right there, that's where the little paleness is in the middle. Okay. Sometimes some of them don't develop well and they become round. Okay, and again, this is going to be a leap of faith. This is going to be a really leap of faith. What they call those round ones, they call them spherocytes, and the spherocytes may be seen in certain diseases. Okay, but anyway, these are the typical looking what you see of an erythrocyte. Some of them you don't see as much because what happens, they might be a little bit twisted or turned and stuff like that, so you don't see it. But when you look at the majority of these, they look very pale in the middle and thicker and darker on the outside, and that's because of these biconcave discs. Okay, and what's the purpose of erythrocyte? To carry oxygen. They get rid of the nucleus. Look, I dare you to look at any of the cells here or any of the cells here and show me and point out a nucleus to me. You can't, because they're not there, with a possible exception. I mentioned that what happens is as we're becoming an erythrocyte, the step before that is called a reticulocyte. What happens is that the red blood cells did start out with a nucleus, but what they do is they start to break it up, like I mentioned, and to get rid of it. They extrude it from the inside of the cytoplasm. Well, what they do is sometimes in cases where people are bleeding, okay, and they're actively bleeding, and when they look at the red blood cells, every so often, what they'll do is they'll see a red blood cell, and that red blood cell will have these little, actually I should have done it this way, little black dots, little dark dots inside there, okay? They'll see these little dark dots, okay? And what those little dark dots are remnants of the nucleus. And since they're not fully adult grown red blood cells, they're called reticulocytes. Reticulocytes. And again, what happens if someone sees reticulocytes on a blood smear, they actually know, and they can actually determine this a couple other ways. But when they look at the reticulocytes, they know that they're bleeding somewhere. Because what happens is the bone marrow's trying to make as many red blood cells. If we're losing blood, the bone marrow is actually that stem cell is starting to make a lot more red blood cells. But what happens when it's starting to make a lot more red blood cells, it can't keep up with the demand. And what the bone marrow does is says, okay, I know reticulocytes, you're not fully adult, you're not mature enough yet, but guess what? We're going to have to send you out. And they do. And as a result, sometimes they get in the circulation. They're called reticulocytes. So that's sort of like, everybody has maybe a couple here and there. They sort of slip out by mistake, But if your reticulocyte count gets very high, people know that they're bleeding, okay? And those are called reticulocytes, okay? Now, a couple of the things that I just want to mention about red blood cells before I pass on to the white blood cells are blood types. I don't know how many people know what their blood type is. I'm a B positive, okay? My wife is an O negative, and the blood banks absolutely love her, okay? They clamor over wanting her to come and donate blood. Now, what does this mean? What do blood types mean? Well, remember we talked about there are proteins in the cell membrane. Remember when we went back to the cell membrane, there are proteins. And some of these proteins are cell markers, okay? And there are special types of proteins that are different, and they actually are on the surface of the cells. They coat the cells, okay? And what happens is red blood cells are coated with a couple of different types of these protein markers, okay? If it's a type O, okay, well, first of all, what happens if we look at blood types, we have A, B, and O. A, B, and O. O means that they don't have those proteins on the surface. There's no protein on the surface, okay? A means that they have an A protein on the surface. B means that they have a B protein on the surface. And AB means that they have both the A and B proteins on the surface. So when they type someone's blood, they look for these surface proteins. If they don't have any of these surface proteins, they're a type O. If they have the A protein on there, which they just use a little reagent and it shows clotting and stuff like that, what happens is they're an type A. If they have the B protein, they're type B. If they have both the AB, they're called an AB. Now, that's the one type. So you say, what's your blood type? I'm a type O, or I'm a type A, or I'm a type B, or a type AB. And now we know. However, they always say, are they AB positive or AB negative? B negative, B positive, A negative, A positive, O negative, O positive. What that means, okay, is also on the surface of red blood cells, there's something called another protein, which is called the RH factor. If that RH factor is on the surface of the red blood cell as that protein, RH factor, then they're called RH positive. Okay, if it's not there, if they don't have that Rh protein on the surface, it's called an Rh negative. Okay, so my wife is like an O negative, which means that she doesn't have. And the thing about what these proteins do is if I have if I again I'm a B positive. Okay, if I get A blood, okay, that if they when they give it to me, my body's going to recognize that that A is not me. And it's going to create antibodies and it's going to destroy the red blood cells. So that's why they always have to match the blood cells with the people. Okay. Now, if you're negative, you can't get something with an Rh positive on there, because if you get if you're Rh positive, that's proteins on there. If you give it to somebody who's negative, what's going to happen is their body's going to say, "Hey, what's that Rh protein doing there? It shouldn't be there," and they're going to create antibodies and destroy it. Okay, so my wife, because she doesn't have any proteins on the surface of any of her red blood cells, she could give her blood to anybody. Anybody could take that. So I'm a B, but what happens if I get O blood as it's called a universal donor. If I get O blood, okay, that means that what happens, there's an O negative blood, excuse me, O negative blood, and what happens is that's fine. Because what happens is there's no protein for my body to react to, and no antibodies are produced, and they're not going to destroy it. Piece of cake. Now, here's one of the problems with females who are Rh negative, okay, which is A negative, O negative, any negatives. Let me get rid of all these. I'm really getting this crazy, okay? Females who are an Rh negative, okay, what happens is when the fetal circulation is totally separate from maternal circulation. The placenta takes care of that. The placenta has a membrane, so the mom's blood never mixes with the baby's blood. They're two separate blood things, because the baby's blood could be totally different as compared to the mom's. So the first baby, what happens is when the baby is born, okay, nothing really happens. Perfect, no problem. The baby's there. But when that placenta separates from the wall of the uterus, there's going to be a little mixing of the blood. So if a baby is an Rh positive, if a baby is an Rh positive blood, what happens is the mom, some of that Rh positive blood gets into the mom's circulation. And what the mom does is creates antibodies. And what these antibodies do is next time that they're exposed to something like that, the antibodies will go crazy, and these certain cells, which are called the lymphocytes, where they make the antibodies in the B lymphocytes, they actually will respond quite fast. Okay? Well, what happens is antibodies are small enough proteins that they're able to cross the placental membrane. So now let's say if I have she has a baby, okay, and what happens is that baby, the body's made antibodies, okay, against, Rh positive. So what happens is now, let's say I have the baby's born, let's say they're an AB positive, a B positive, an A positive, an O positive, because positives are a lot more common than negatives. What's going to happen is the antibodies that mom made from that first baby, who's long and now is becoming, terrible twos or something like that. What happens is now the antibodies could cross the placenta and go to the red blood cells of all these red blood cells in the fetus and destroy it, and the baby may die. Okay, so what they do is, in moms who are Rh negative, around the time of birth, either a little bit before, at the time of, or just shortly after, within a very short period of time after, they give them a drug called RhoGAM. They give them an injection of this RhoGAM, which actually stops the antibody production against the Rh factor. Okay, so they won't have a problem of having a baby that's aborted because of antibodies crossing the placental membrane later on down the road. But that's just a little about blood types. And this is something that I want to show you, because what happens is hemoglobin is pretty stable. However, we talked about the genetic code, and we talked about gene expression. Okay, and gene expression does what? It codes for making a specific protein. Hopefully you know that by now by looking at the DNA. Okay, each gene codes for a protein. Well, in some cases, there's been genetic mutations. And this is the mutation, and this mutation right here is called sickle cell. And what happened in sickle cell is that at some point during one generation to another or something like that, there was a mutation in the hemoglobin which changed one amino acid in that amino acid chain of hemoglobin. Very simple. One code goes wrong. And as a result, the hemoglobin isn't perfect. And when there's low oxygen tension, like in the air, like higher altitudes, stress, airplane travel, and stuff like that, what it does is the protein, the hemoglobin protein, which is now sickle cell hemoglobin, which is not as stable, starts to unwrap itself. It unwraps itself and unboxes itself from the typical box. As a result, what happens, it causes an actual functional change in the cell. And these are called sickle cells. That's a sickle cell, 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 there's a sickle cell up there, we see them over here. These are all sickle cells. They start out looking like a red blood cell. But when the oxygen levels are low, what happens is these cells, because the hemoglobin is unstable, unwraps itself and causes a change in the shape of these red blood cells. They look like sickles. Okay, so what's the problem? Well, red blood cells are red because they're sort of like little taco shells or like burrito shells. They can actually fold to get through vessels and stuff like that, and they're very pliable. Sickle cells are a little bit different. Because the red blood cells are round, they're able to sort of like roll through smaller vessels. There's little small vessels called capillaries and stuff like that, where the sickles, because they have points, they catch. And when one catches, another one catches on the first sickle. And then another one catches, and then another one catches, and then another one catches. And all of a sudden you have all these sickle cells that sort of start to bunch up on top of each other as they go through a vessel. What that does, it causes a clot, decreases blood supply. And the problem that we get with sickle cells are things like strokes, ulcers, a loss of limbs and stuff like that, simply because now a vessel gets blocked by these sickle cells because they don't slide through the vessels nearly as easy as a round, regular erythrocyte. And that's what sickle cell is all about. Abnormal protein, simple mutation, very simple mutation, minor change, but that minor change makes a big difference in the stability of that hemoglobin molecule, which now unwraps itself and causes a change in the shape of the cell. And that's what we see in what was called sickle cell anemia. Okay, so that's a little bit about sickle cell, and I just want to bring that up to show you what that is, because when we do in the lab, there's a sickle cell slides. Some of them are okay, some are hard to find the sickle cells, but they're there. Okay, and again, because it's a genetic mutation, African American. There's other types of what are called hemoglobinopathies, which are also minor mutations of the hemoglobin. They're called thalassemias and stuff like that, and there are different groups of people that will have that. Okay, so anyway, that's a little about sickle cell, okay, and a little bit about those red cells. Now let's talk about white blood cells, okay? Now we have a bunch of different types of white blood cells. Now I love this slide over here because it has all the white blood cells I ever want to know, okay? This one right here is called a neutrophil, okay? See those little granules in there, but they're sort of neutral? Here's that eosinophil we talked about because it has red granules. Here's that basophil because it has purple granules, okay? This one right here is a lymphocyte. Okay, and I'll tell you why it's a lymphocyte in a second. And this one right here is called a monocyte. And I'll tell you why that's a monocyte and why these two, they look sort of similar to each other, but I could tell that this is a lymphocyte and this is a monocyte, and I'll tell you about that in a second. This one up here, okay, is called a band, B-A-N-D. And I'll talk about what a band is. A band is an immature neutrophil. Okay, so we look at here's the neutrophil here, here's a lymphocyte, here's an eosinophil, here's a basophil, a monocyte, here's a basophil, and then, that's what we see there. Okay, and let me talk about each of those individually and talk about the importance of each of those. Now, first of all, let's talk about what the cells are. Neutrophils, okay? Neutrophils, like we showed in an earlier slide, about 50 to 70% of my white blood cells at any time are neutrophils. Neutrophils also go by a couple different names, okay? So we call them neutrophils, or something they call them polys, or PMN. PMN stands for polymorphonuclear. Now, if you remember what the neutrophil looked like, the nucleus had lobes to it, Okay, here's the cell, and the nucleus had all these different lobes to it, 3 to 5 to sometimes more lobes, and that's what they call a polymorphonuclear lymphocyte or leukocyte because it's a white blood cell, but it's a PMN, a poly, or a neutrophil. The reason why it's called a neutrophil is because the granules stain very neutral. They're very pale. Okay, you have to look really close. This is the one that's the most important in bacterial infections. So when people have have bacterial infections caused by bacteria, this one goes up. Okay, now there's a word that I'm going to tell you right here that is going to be interesting. Okay, if you're in the hospital and they say they have a shift to the left, neutrophils on the left side of this chart, what happens, that means that the neutrophil count's gone way up. When people have an infection, what happens is when they look at their CBC, which everybody knows is, their white count goes way up. But then they also do that differential, and when that differential goes way up, okay, in regards to neutrophils, they're pretty sure it's going to be a bacterial infection. So that's what we usually find with large numbers of neutrophils. Okay, lymphocytes. Lymphocytes are the second most common, and what we find is this is between about 20 to 40% of my white blood cells are going to be lymphocytes. Again, my polys are about 50 to 70. My lymphocytes are about 20 to 40. Okay, and the lymphocytes are there. They have three types of lymphocytes. Okay, and I'll talk about those in a second. But what happens is we see these in viral illnesses and mononucleosis. What happens is sometimes they do a mono test. If you ever had mono, they've done a little blood test to look at the monos and stuff like that, look at antibody titers, okay, which is called heterophile antibodies. But they could also sometimes just look at the peripheral smear, and they have lymphocytes, but the lymphocytes, they don't look right. They look different. They're called atypical lymphocytes, okay? Also, some types of leukemias and Hodgkin's will have large numbers of lymphocytes, okay? So the two most common, polys, 50 to 70%, lymphocytes, 20 to 40%. I'll talk more about each of those. Monocytes. Monocytes are basically eaters. Okay, they're indiscriminate eaters. We see them in viral illnesses and stuff like that, herpes, cancers and stuff like that, rheumatoid arthritis. The number, again, the number is probably less than 6, 7, 8%, 9%. They're a relatively low number of monocytes, but they're eaters. They're indiscriminate eaters. Okay, so those are monocytes. The fourth type we get right here are called eosinophils, and those are the ones that have red dots in them. Okay, and where we see the eosinophil count go up, two things basically, allergies and parasites. When people have parasitic infections, the eosinophil count goes up. People with allergies, their eosinophil count goes up. If you ever see the commercial, they talk about asthma, basically, you have asthma and a high number of circulating eosinophils, okay, you may be subject to using a certain type of a drug, for the asthma. Well, guess what? Most asthma is actually triggered by allergy. So there's no big whoopee there. I mean, it's not like, oh my gosh, this is something, it's made my week or something like that. But what happens is eosinophils are higher in the body in people with certain allergic responses as well as in parasitic infections. Basophils. Basophils are basically there because of inflammation and stuff like that. And when the dark granules basically are there because what happens is one of the common things in the dark granules is something called histamine. And when the body tries to heal, it has to heal by creating inflammation. Inflammation is actually a healing process. Okay, so to do that, these basophils will go in there, and what they do is they start to release this histamine, and the histamine causes blood vessels to dilate. You ever notice, when you have an allergy or hives, it turns red, it's swollen, and it's itchy. Okay, and that's because the histamine's relieved. What do you do to relieve it? You take in antihistamine because that decreases those basophils releasing the histamine. It works against the histamine. Okay, so these are a little bit basically things about that. And again, we talked about that word, that differential. That's that differential that we talked about. Okay, how we look at the different percentages and stuff like that. Don't worry about this. This is something that a lot of people in the hospital use to actually look. It's a fishbone grid. They put the platelet count, the white blood count here, the hemoglobin, hematocrit, and it just gives them an idea about what the overall CBC says. Okay, now let's talk about polys. Here's a poly. Okay, now let's look at that nucleus. Okay, here's a little lobe here, here's a little lobe here, there's something there, there's something right there. If you look at the granules are very, very faint. Okay. Again, this is about 50 to 70% of my white blood cells are going to be polymorphonuclear leukocytes or neutrophils. Okay. And this is a neutrophil. You see how the nucleus has multiple lobes? You know, they're usually connected by a little strand. Okay, a little strand. You see a little strand right there? Let me get rid of my little drawing so you can see little strands will connect all those. And they have multiple lobes. Okay. And those are called nuclear leukocytes. And the most common cause is probably bacterial infection. When somebody has a bacterial infection, that's what causes these polys to go up in number. Okay, and again, I mentioned shift to the left. Shift to the left. And instead of having 50 to 70%, now they have 85%. They have a white count. The normal white count is between 5 and 10,000. Now all of a sudden they have a white count of 20,000, and 80% of them or 85% of them are neutrophils. Guess what? Boom! It's a bacterial infection most likely. Okay, so that's what we see with that. Those are called neutrophils. Basically, and these are sort of interesting. What they do is they actually migrate to the area. When somebody has an infection, what happens is certain chemicals are released, which are called chemotactic type chemicals, and they actually attract these white blood cells to go to that area to fight the infection. And the white blood cells, then they have a blood vessel. This is sort of actually cool. Blood vessels are like this. What that happens is normally they're just swimming down the middle and stuff like that. They're floating just along in the flow inside the blood vessel. What happens is they actually migrate to the surface of the blood cell, or the surface of the blood vessel, and they start to roll down the wall. And they finally get to the area of the infection. They actually go through the wall and to be able to fight the infection there. They're eaters and stuff like that. They release all kind of chemicals that will release things. Okay, so those are called polymorphonuclear leukocytes or neutrophils. Okay? And now this right here is called a band. Now this is a poly. You can see a poly there, there's a poly there, there's a poly there. But this one right here, instead of it being segmented, it's just sort of like a horseshoe or a band. Okay. And what a band is a band is an immature neutrophil. Okay, it's not quite ready. The nucleus hasn't segmented as much, and as a result, it looks more like a band as compared to a segmented nucleus. And as a result, because they're immature, they don't work as well. You know, they're not like an adult grown up type of a neutrophil, so they don't work as well. And because of that, you know, they're there. So what happens is when we see that shift to the left, okay, and we see an increased white count, what you're always probably going to see is an increased number of bands. Because what happens is the bone marrow is putting out as many neutrophils as it can to fight the infection. But if that infection keeps on going, what happens is the marrow says, you know what? You know, I don't have any more on the shelf right now, but I do have some in waiting. I have some bands that I could send to you. And they send the bands out instead of the neutrophils because there's no neutrophils left. Okay, and so that's why those band numbers will increase in infections as well. Okay, those are called neutrophils, and those are bands. Okay, now here's a lymphocyte. Okay, and your lymphocyte. Now I talked about a lymphocyte and a monocyte. Okay, the difference between a lymphocyte and a monocyte in regards to appearance, monocytes are bigger. If you look at a lymphocyte, the lymphocyte's almost the same size as a red blood cell. Okay, it's very close. Monocytes are going to be a much larger cell. Okay, monocytes will have a very large nucleus. Okay, one of the things we see about lymphocytes is the nucleus seems to be sort of eccentrically located. Here's like a rim of cytoplasm around the side. Okay, very large nucleus, and they're about the size of a white blood cell. Okay, now here's the interesting thing about lymphocytes. Okay, they're increased in like viral infections and stuff like that. There will be other infections, but again, what happens is I have three types of lymphocytes. Okay. And this is sort of a cool thing. If you read it like immunology, this is where to go. Okay, you have three types of lymphocytes. You have one type of lymphocyte which is called a T lymphocyte, another one which is called a B lymphocyte, another one which is called an NK lymphocyte. Okay, now what happens is B lymphocytes, like I mentioned before, will mature and they actually become what are called plasma cells. Plasma cells, and these plasma cells make antibodies. Okay, and the antibodies will fight off antigens. What an antibody is, it's a protein that the body makes to be able to destroy an antigen. An antigen is a foreign invader. Okay, so this is called humoral immunity because it's using antibodies. T lymphocytes also have a little bit of, and these B lymphocytes have a memory. We talked about with the Rh factor before. What happens, it's the B lymphocytes that will remember that Rh factor protein that they got in by mistake to an Rh negative mom, and has a memory. And so what happens is when they're exposed to it the next time, they can mount a faster response. Okay? T lymphocytes create what we call cellular immunity. Cellular immunity, okay? And basically, they go in, they start to, and what happens is they actually work with the B lymphocytes in the immune system to try to make both cellular chemotactic to destroy things as well as antibodies to work together. Okay, I'll get back to that in just a second. The third type is called an NK lymphocyte. NK stands for natural killer. NK lymphocyte. And basically they're indiscriminate eaters too. They just go in and kill stuff. Now, one thing I just want to mention about T lymphocytes and B lymphocytes, okay? They both have to work together in this immune system. They're both part of the immune system, okay? And they both have to work together, okay? What happens is I have my B lymphocyte over here, okay? Now, when you look at lymphocyte underneath it's really difficult to tell the difference. When you look at them under the microscope, they all look about the same, okay? But my B lymphocyte is making an antibody. My T lymphocyte is basically doing a cellular type of a destruction, okay? What happens is these two have to talk to each other. They need to talk to each other, okay? You're wondering where this is going. And this is quite important. What happens, there's another type of a lymphocyte. It's called a CD4 lymphocyte, CD4. Okay, you say, oh, well, that's just another lymphocyte, you know. The T lymphocytes are actually CD8s, and this is a CD4 lymphocytes. And these CD4 lymphocytes are called helper Ts. So they're basically a T lymphocyte that actually helps to influence the B lymphocyte, so the B lymphocyte and the T lymphocyte could work together. Okay, so they're called helper Ts. So this is called a killer T. And the CD4 cell's a helper T. Now, you're saying more worthless information. And I'm going to tell you you're wrong. Everybody's probably heard of this. HIV. HIV is a virus, as we know. It's a retrovirus. It's an RNA virus. And what HIV does, it really likes the CD4 cells. It really affects the function of the CD4 cells. Therefore, if the T's can't communicate with the B's, the immune system ends in the dumper. It doesn't work. When people have HIV and they get AIDS, HIV is basically the virus, okay? And people go many years with that are HIV positive before they actually develop AIDS. And the AIDS is basically they start to show what? Acquired immune deficiency syndrome. And why is it immune? Because these CD4 cells are low in number and they don't work. We don't have a communication between the T's and the B's, and our immune system is really horrendous. It can't fight infections. When people have AIDS, what do they die of? They die of two things. Infections that can't be controlled, because again, the immune system is trashed, or unusual types of cancers. And that's also affected by this immune system. There's one called a Kaposi sarcoma. If you watch the thing with Tom Hanks, the movie with Tom Hanks, he started to develop the blotches. And that's from Kaposi sarcoma, which sometimes it occurs in people from Mediterranean areas just normally, okay? And Kaposi sarcoma is like a vascular type of a tumor, vascular type of a cancer. What happens is also one of those things we see in HIV. They get Kaposi sarcoma and die of Kaposi sarcoma. So Kaposi sarcoma or unusual other cancers or infections. There's an infection, it's called pneumocystis carinii, which is basically a bug that's in our lungs, and we all have this, but we keep it under control. HIV, they can't. And this bug goes crazy, and they get pneumocystis pneumonia and die from pneumocystis pneumonia. Why? Because the lymphocytes, my CD4 lymphocytes, don't work. That's what HIV likes. And the way a virus works, I've probably mentioned this before, is the virus is basically a piece of RNA or DNA. Okay, and what's RNA or DNA? Has a code for a protein. Okay, and that virus is encapsulated in like a protein coat around the outside, a shell. And what happens? It gets close to the cell, and the cell says, that shouldn't be here. Let's eat it. They eat it up. But once they get inside the cell, those lysosomes start to melt away that protein shell. It releases the RNA or DNA. If it's an RNA, it's a retrovirus. If it's not, it's a DNA virus. Releases that, puts it, and that RNA or DNA goes to our own nucleus, gets inside the nucleus, and incorporates itself in our own DNA. And what does it do? It makes an abnormal protein, because that protein is something that's abnormal, and that's what causes disease. And that's what happens. Okay, so anyway, we can't fight it with HIV/AIDS simply because what happens is CD4 cells can't allow the T cells and the B cells to communicate. They're two separate worlds. Okay, so they're totally. This one doesn't talk to this one. You know, siblings that don't get along anymore. Okay. Now look at this one. This one has a large nucleus. It's eccentric, but what happens is if you look at the size of the cell is a lot larger. This is a monocyte. That's a monocyte. And monocytes are basically eaters, they're killers, okay? And they do a lot of other things. In other words, when we talk about the bone actually has certain cells that are called osteoclasts that come in to eat bone and destroy bone so new bone could be laid down, like at fracture or remodeling or normal everyday maintenance. What these osteoclasts are is basically they're modified monocytes. They have all these enzymes in there to sort of destroy things. And it's called a monocyte. Okay, very few. This one right here, okay, is a basophil. Look at those granules. Those granules are dark purple, and those are the ones that have the histamine. Here's a basophil right there. This one up here is an eosinophil. Eosinophil has red granules, and we talked about those are there when we see with parasites and allergy. Okay, look at the size. Here's a red blood cell right here. Okay, here's another red blood cell. Here's an eosinophil. Here's a basophil. There's a lymphocyte. What can I tell me about the lymphocyte? The cytoplasm is almost totally taken up with the what? The nucleus. Okay, and that's important because again, when I'm making antibodies are proteins, and I have to make the proteins by changing the DNA code in the nucleus on a B lymphocyte. But they're also about the same size as a red blood cell. Makes sense. Okay, so that's what we see. So we have a basophil, we have eosinophils, and we have another neutrophil or lymphocyte over here. Okay, now I put this in here because I just want you to look at this for a second. What's wrong with this picture? Something that we talked about at the very beginning. We're talking about red blood cells, they're in the millions. White blood cells are in the thousands. I can see my red blood cells in there. What are all those purple things? Those are white blood cells. Look at that ratio of red blood cells to white blood cells. What can you tell me? That's wrong. There's far too many white blood cells, and this is what we see in something like a leukemia. So you see too many white blood cells, and a lot of these white cells don't look like they're mature. There's acute leukemias where we have immature white blood cells. This is an important factor as well. Let's think about this. If we think about white blood cells are very important in fighting infection, people with leukemias, guess what they also die of? Infection. They have a significant risk of infection or bleeding. Okay? Well, you say, well, they have so many white blood cells. Why can't these white blood cells fight the infection? The reason why is what happens in leukemia. They make lots of white blood cells, but none of them ever get to be adults. They all stop. In acute leukemias, chronic leukemias, they do get to be adults, but in acute leukemias, which are very common, they stop at a certain point. Therefore, these white blood cells are out there. They're populating the air, but they don't do anything. They're worthless. They're taking up space. They actually increase the viscosity of blood. The blood becomes a little bit thicker, which people are a little more likely to develop clots and all kinds of other problems with this as well. So the problem with leukemia is we have lots of white blood cells, but none of them are working because they're all immature. They're not developed enough in many cases to get to a point where they're actually mature. That's in acute leukemias. Chronic leukemias, we have older, mature, but they just don't work as well. Okay, but I just threw this picture in there because it showed a leukemia. Okay, now one of the things I didn't show before were platelets. Okay. And if you look here, see all these little clumps of stuff right there, clumps of stuff? That's a big clump right there. There's a poly right there. There's a lymphocyte, okay? But you look at all these little, that's not dirt on the slide. Those are all little platelets, okay? And the platelets, like I said, are important in clotting, okay? When I have a hole in a vessel, because the platelets are sticky, they plug the hole. They're actually when the inside lining of the vessel is exposed to the blood, it actually creates a chemical signal that actually draws the platelets to stick to them, and as a result, these platelets then cause a plug. So that if I have a hole in a vessel right there, the platelets start to stick right there, and they actually cause what's called a platelet plug. Then what the platelets do is they release a bunch of chemicals. These platelets then release chemicals, thromboxane A2 and a bunch of other stuff. And what those chemicals do is they actually go to the red blood cells and say, hey, listen, we're having a party, come on over. And all of a sudden these red blood cells start to congregate, and what the red blood cells do, and they congregate, that's what we call a clot. Okay, so when the platelets are actually what starts it, they form a plug, then they release chemicals, the chemicals draw red blood cells form, and they create a regular clot. Okay, I can't think that aspirin, because it decreases the stickiness of a platelet, it's not really a blood thinner or an anticoagulant. They say blood thinner, that's a bad word. It should be anticoagulant, something, Coumadin, heparin, Lovenox, all these are anticoagulants. But platelets or aspirin is not. It's basically something that just decreases the stickiness of platelets. That's all it does. It does nothing else. It just decreases the stickiness of platelets. Okay, I could go into another half an hour about that, but I think we're about ready to try to wind this puppy up. Those are platelets, okay? And this is basically what we're talking about. That what happens is when you have an injury, what happens is the red blood cells, first of all, the platelets stick to the wall. And then the platelets then release chemicals, and the chemicals will draw the red blood cells. And then when that actually it's interesting, because what happens is there's called a clotting cascade. Okay? It's sort of like if I had a line of dominoes lined up, what happens is if I push that first domino over, that second domino is going to fall, which then pushes over the third, and the third one falls, and the fourth, and the fifth. And this is what happens. What happens is once those chemicals are released, it starts this what's called a clotting cascade, various factors, okay? And one factor actually causes an activation of another factor, which causes an activation of another factor, which causes an activation of another factor, and that's what we call, again, that clotting, that coagulation cascade. And basically what that does is eventually that allows that clot to form and unite and create a clot. When people have hemophilia, which is, fortunately not that common, but and again, we saw that like royal families. Why? Because it was inbreeding. They didn't want anyone else to get in there and rule the country or anything like that. So what they did is they inbred all these people, and what happens? They passed on this hemophilia to a lot of the offspring. And what happens is the hemophilia means that they don't have a certain factor, or one or two or more factors in this clotting cascade. And as a result, what happens is they don't clot, because one, it's sort of like taking this picture on the bottom, but let's say remove a couple of these. So even if this one falls, it's not going to knock over the third that's there. Okay, so that's what happens with hemophilia. Okay, and then again, when people get anticoagulants, it's basically sort of stops that. It sort of like prevents that cascade. It affects the cascade in different spots according to which type of anticoagulant someone takes. Okay, so that's just a little about clotting and coagulation and stuff like that. It's an important factor that we have. Some people are more hypercoagulable. Females who are on birth control pills are more likely to have clots. People who smoke are more likely to have clots. People who have cancers are more likely to have clots. People who are inactive are more likely to have clots if they're sitting in one spot for a long time, like long car rides, plane rides, stuff like that. They're more likely to have clots and stuff like that. We'll talk about those later in the semester. Okay, about that stuff. But that's just a little about coagulation. Okay, and again, we're talking here. Here's some more neutrophils right there. That's actually more close. That's just that's almost a band at this point. Because it's starting to segment. You see a segmentation right there. So it's almost that point. This is definitely, what's that? That's a lymphocyte. Okay. What do you think that is right there? That's an eosinophil, obviously. What's that? Well, it looks like a lymphocyte, but it's too big, a lot bigger. That's a monocyte. What are those? Those are platelets. So what they do. What happens is a lot of times what they'll do is they'll do what's called a peripheral smear. They'll just take a little bit of blood, put a little drop on there, and they slide this cover slip over the top, and it stretches it out really thin. And then they could look at it. They could stain it and look at it at the microscope. And they do what's called a right stain, and they're able to determine the colors. That's what stains those granules, stuff like that. So anyway, that's an important factor about blood. So hopefully this gives you a little bit better idea about what blood's all about. Okay. Again, it has multiple functions. It has a function in regards to being able to carry oxygen, which is basically the object of the red blood cell, fighting infections, responding to viruses, and all kinds of stuff like that. These are all things that the blood does. Hopefully, this gives you a little bit better idea about what blood cells do, so you could actually understand them just a little bit more. We know that they're part of connective tissue, but I really thought when I finished connective tissue that we probably should spend a little bit more time. I didn't think about this, and you might not even get to the end of this. I think it's sort of interesting, but I thought maybe blood deserves a little bit more attention than we actually gave it. So I thought it was a good idea to do this, and this is sort of like a late add-on. So, but it's there if you want to watch it, and we'll go from there. Again, as always, if you have any questions, make sure you ask. And if send me an email or ask during one of our WebEx sessions or something like that, and I'll be more than happy to answer any question. I love questions again. Makes things interesting. And there's always questions, and if you don't want to ask it in front of everybody else, you can always, like I say, email me. In the meantime, though, be safe and be healthy, and we'll see you with our next one.